Anyway, it's 11 o'clock now, so we're gonna just get rolling, and if a few more people show up over the next few minutes, then that's great. They'll just kind of miss the housekeeping bits. But for anyone who doesn't know me, my name's Lauren Benoit, and I'm a market development agronomist with Bayer in southwestern Ontario. So I cover London up to Kincardine, roughly, and I'm really in kind of the corn rootworm hotspots. I cover most of the corn rootworm hotspots of the province. So we've been doing a lot of work with SmartStax PRO and a lot of root digs. And a lot of root digs and everything to get that done. So today and this week, we've been creating and generating a lot of content around that to kind of, do a lot of education bits and whatnot. And this is one of those things. So today with us is Dr. Edwin Benkert from Iowa. So Edwin is a technology development rep focused on seeds and traits out of Iowa, and he's going to be talking about his experience with SmartStax PRO today, and for about 20 minutes, and then I'll be asking some questions as well. And then if any of you have questions, feel free to throw them into the chat, and then we will touch on them at the end. So with that, I will let Edwin take it away.
Yeah, thanks, Lauren. You guys, are you able to see my slides here?
Yep, I can see your screen.
Okay, perfect. So thanks again, Lauren, for asking me to do this. My background is in entomology, so my PhD, that's what it's in. And while I was getting my degree, I worked on corn rootworm management and BT resistance. So kind of right up my alley. But today I'm going to talk about SmartStax PRO technology using RNAi for corn rootworm management. And before we get into it, slides aren't advancing. Okay, did they advance on your end?
Yeah, that worked for me.
Perfect. So before we get into SmartStax PRO, I thought it's a good idea just to quickly review corn rootworm, just to make sure we're all on the same page. So there's two species that cause injury to corn that we're most concerned about. The first one's going to be that western corn rootworm, which are the yellow beetles with the black striping. You can see that here on the right. And the second species is going to be the northern corn rootworm, which are tan when they first come out of the soil, but then they take on almost like a emerald green color after they feed on that corn leaf tissue. Both species are very similar in their life cycle. So they both overwinter as an egg before hatching in the spring to feed on corn roots in the soil as a larvae, before pupating and then emerging from the soil as adults, kind of mid to late summer, before they lay their eggs back into the soil. So both species very similar. Both species also only have one generation per year. So they, unlike some of our other pests like western bean or corn earworm, where we could see multiple generations per year, corn rootworm is only going to be one generation. There are two exceptions to this. The first exception is, this one's probably more common in the US, but it's called the Western corn rootworm variant. Was first found in northern Illinois. It's kind of spread a little bit from that, but basically the Western corn rootworm variant, it still has one generation per year, but instead of laying its eggs back into corn like the normal life cycle would be, the variant's going to lay its eggs at random anywhere. So if it happens to be a soybean field that's going to be rotated back to corn the following year, you could see some injury in that first year cornfield. Like I said, more of a U.S. problem, the variant is, but then other life cycle adaptation is going to be the northern corn rootworm that exhibits extended diapause. And so in this situation, diapause is going to be that overwintering period as an egg, and populations of northern corn rootworm that have extended diapause, instead of hatching every year, they're going to hatch every other year. And so, again, kind of an adaptation to get around crop rotation, but northern corn rootworm with the extended diapause, it's probably a little more common in Canada, occurring in some pockets there. Uh, there, so probably a little more familiar with that one. Like I mentioned, the larval stage is the one that we're going to be most concerned with. The larvae will cause damage by feeding on the roots in the soil, and we rate that damage using a 0 to 3 nodal injury score, which we can cover on the next slide. And adults can cause damage by feeding on silks or clipping silks back to interfere with pollination. But unless you have a really heavy, high pressure situation, it usually this isn't that economical. It's not that common. And so when we're managing adults, normally we're focused more on reducing the number of females that are going to be laying their eggs for next year. So reducing populations that could be an issue next year. So that 0 to 3 nodal injury score, this is the way we evaluate how much damage the larvae are causing to the corn plants and therefore how much yield we can expect to lose. So with the 0 to 3 score, if we look at a root system, roots grow off a corn plant in these nodes here. So they grow off in these circular nodes around the stalk. And when we're evaluating root feeding, we're going to be focusing on nodes 4, 5, and 6. And we're going to be looking for pruning or feeding here that is 3.8 centimeters or less from the tip of the feeding to the base of the stalk. So if that feeding is 3.8 centimeters or less, we count that as having been fed on. And we'll go through and we'll count the number of roots that meet that criteria on that node, and then divide that by the total number of roots on that node. We do this for nodes 4, 5, and 6, add those numbers together, and we get a number, a total number between 0 and 3, where 0 corresponds to a healthy root system, where there's no feeding, and a 3 would correspond to approximately 3 nodes having been fed on within that 3.8 centimeter criteria. And this is important because, with root feeding, you can get a yield loss up to 45%. And so, universities in the States and elsewhere have found that, a score of 1 on that 0 to 3 scale is going to correspond about a 16% reduction in yield. A 2 is about 30, and a 3 is 45%. So that's where they're getting that 45% number. And the other thing to note with this is that's just from the feeding caused by the corn rootworm. So this doesn't take into account, environmental factors such as drought, fertility issues, disease, some of those other things that could happen in any given year. This yield reduction is just from the physical feeding on the root system. So covered corn rootworm, I always like to review our BT traits as well before we get into SmartStax PRO. So, when it comes to corn rootworm management, we really only have 4 BT proteins that are actually active against corn rootworm. So, we have a lot of different trade names, but at the end of the day, there's only 4 proteins that are serving as our modes of action there. And you can see, the different combinations that those proteins are in here. We have our SmartStax, then our Corteva trait packages, and then finally our Syngenta trait packages down here, and the corresponding proteins that make up those. So initially we released proteins as a single trait, where we would have 1 protein present within that hybrid. But then we began pyramiding proteins together in different trait combinations, just to get more than 1 mode of action present within a hybrid there for better management of corn rootworm. So the way these BT toxins work is, you remember those proteins are insecticidal proteins. So they're naturally expressed by the soil bacterium BT Bacillus thuringiensis. And what we've done is we've isolated the gene sequence in that soil bacterium that expresses those proteins, and we've inserted that into the corn plant so that those proteins are expressed in the root tissue where the corn rootworm are going to be feeding. And so where these proteins are working and actually killing the insect is going to take place in the midgut. And so if you think of insects, the midgut is similar to like the small intestine in animals. So where a lot of our nutrients are absorbed from the food that we eat, and where you're getting, the actual materials you need from whatever you're eating. So that Bt protein is eaten. It enters the midgut where it becomes activated. And so once that protein becomes activated, it's going to bind to receptors on the cells that make up the wall of the midgut. So, the wall of the midgut separates the gut contents from the blood of the insect, and that's made up of a bunch of different small cells. So when this activated protein binds to the receptor on those cells, it causes pores to form in those cells. And once those pores form in the cells, you get gut fluid rushing into the cells, and then it becomes like a water balloon when you have too much water. The cells start to burst. You get enough of those cells that begin bursting, and eventually you get pores and holes forming in the midgut cell wall and cell lining. And once you get holes within that midgut, that's where you get gut contents mixing with blood and vice versa. The insect develops a stomach ache and ends up dying. So this process is pretty quick. With BT, it's usually about 10 to 12 hours from ingestion to death of the insect. So that's how BT works. The other thing to keep in mind with our BT traits and proteins is we do have resistance confirmed, by our academics to our proteins. So Western corn rootworm has resistance confirmed to all four of those proteins in certain areas of the corn belt. And northern corn rootworm has resistance confirmed to two out of those four proteins within the corn belt. So, with resistance, keep in mind it occurs in pockets, right? So it's not, we don't have Western corn rootworm resistance to all four proteins all over the corn belt, right? It's specific populations and specific fields in pockets where this is occurring. So it's not widespread. And it's not, we don't have populations where all four proteins are no longer working. That's not that common, right? So in a lot of cases, we may have resistance to, say, cry3bb1, but cry34, 35 is still working, or similar with our other proteins there. So the take home here is, our BT traits are still very effective in most areas of the Corn Belt. So they still work very well, and they're still very effective at managing rootworm. But we do have to keep in mind, resistance is out there and has been confirmed. So we have to make sure that we're managing the rootworm properly and using these traits properly so that we can make sure that they keep working. So now we'll get into SmartStax PRO. So SmartStax PRO technology with RNAi is going to be the first product on the market that uses RNAi for implant insect protection. It's also going to be the first product on the market that has 3 modes of action for corn rootworm control. And so what sets SmartStax PRO apart from our previous SmartStax hybrids is, we still have the same above ground modes of action in SmartStax and SmartStax PRO. We still have the same herbicide tolerance in SmartStax that we have in SmartStax PRO. The difference is going to be when it comes to our below ground protection. So in SmartStax and SmartStax PRO, we still have the cry3bb1 and cry3435 BT proteins. But in SmartStax PRO, we're bringing in this new unique mode of action, RNA interference, where we're targeting a gene sequence within the rootworm called DV-SNF7, DV-SNF7 or DV-SNF7. So this third unique mode of action is what sets SmartStax PRO apart from our SmartStax hybrids, as well as from our competitors, because it's a new non-BT mode of action. And so, how is RNAi different from the way BT works? So you remember BT is happening in the midgut, interfering with the cell lining of the midgut. With RNAi, we're affecting the way DNA is producing proteins within the corn rootworm. So in a normal situation, we have DNA here, and everything has DNA, right? Rootworm have DNA, the corn plant has DNA, we have DNA. But DNA is just the instruction manual. So, it's got all the instructions for everything that needs to happen in the body, but it's just an instruction manual. So we need mRNA to come in and read those instructions. And then the mRNA is going to turn around and make proteins based off of those instructions that the DNA is providing. And these proteins are going to go out, and they're what are actually responsible for all the functions in the body: growth, metabolism, movement. Everything that happens within the body happens because of these proteins. So that's the normal process there. When we introduce RNAi that's present in SmartStax PRO, we still have the DNA that's acting as an instruction booklet, and we still have the mRNA that's coming in and reading those instructions. But we introduce the RNAi that's present in SmartStax PRO, and we target that mRNA, and that causes the mRNA to become broken up. If the mRNA is broken up, it can't turn around and produce any proteins, and without those proteins, we get death in the insect. So, a couple things to note with the RNAi and SmartStax PRO, we're targeting that gene fragment DV-SNF7. And if you think of, gene fragments and gene sequences within the body, right? There's certain gene sequences that we could live without, right? There's some that if they're not expressed, we could still go on living and everything would be fine. And the same is true with the rootworm. There are some gene sequences and gene fragments that are called major housekeeping gene sequences. And these major housekeeping gene sequences are essential for life. So without these being expressed, life doesn't happen, right? And so that's what the DV-SNF7 gene sequence is in corn rootworm. It's one of those essential housekeeping gene sequences that absolutely has to be expressed in order for the rootworm to survive. And so that's what we're targeting with the RNAi and SmartStax PRO. We're preventing that from being expressed, preventing those proteins from being produced, and causing death to the insect. The other thing with RNAi that sets it apart from BT is with BT, our kill time is around 10 to 12 hours, so pretty quick. With RNAi, the kill time is a little longer. It's about 4 to 6 days. And the reason for that is because even though we're not producing any new proteins here, we already have a supply of proteins that were produced previously that have to be used up first before the insect will die. And so that length of time is usually about 4 to 6 days before that happens. So that's SmartStax PRO. That's how RNAi works. I've got some data that we can talk through from some trials that we've done in the US the last couple of years in the Corn Belt. You can see the locations of the trials here within the Corn Belt in the US. And we divide the locations up based on corn rootworm pressure using that 0 to 3 nodal injury scale on an unprotected double pro hybrid. And we bucket them into no pressure, low pressure, medium to high, and then very high pressure. And you can see how those are distributed, on the map here. So when we look at the root injury, so we had the 69 locations over 2 years that had root injury. And on the y-axis here, you remember this is our nodal injury score. A 0 is going to be very good, healthy. That's what we like to see. Whereas the higher we go, that's going to mean more root injury. And we can see when we break it down, that double pro is not going to have any protection against corn rootworm. So this would be our check. You can see it's about a 1.4. So at least a 17% yield reduction from corn rootworm feeding happening here, and pretty high pressure in this data set. When we add a soil applied insecticide on the double pro, we do see a significant reduction in root injury there. And then when we compare our two competitor trait packages to the Double Pro, we also see that they are reducing root injury significantly, but not significantly more than our SmartStax trait packages. So SmartStax is reducing root injury significantly more than a soil applied insecticide or Duracade and Chrome from our competitors. And then finally, when we look at SmartStax Pro, we see very good performance and protection with it significantly less than even our SmartStax trait packages in this data set. So a couple things to keep in mind. In the United States, the EPA, Environmental Protection Agency, has set 0.5 on that 0 to 3 injury scale as our threshold for root injury on a traded product. So if we have a traded product that has root injury above that threshold, then that requires us as a company and our competitors to go out and assess what's going on in that field, figure out if what's going on. Maybe it's decreased trait performance, maybe there's higher populations, but we have to figure out why the injury is higher on that traded product than our threshold. And so I point that out because even though SmartStax is providing very good protection here, it is above that threshold, at least in this data set. And the other thing I point out is, SmartStax Pro is providing very good performance and very good protection, but it's not perfect, right? It's not zero. We're still seeing some injury at these locations in this data set. And so, with SmartStax Pro, it's a very effective tool, right? It has very good protection and performance, but it's not a silver bullet. We still need to be keeping in mind those other management tactics and other tools in the toolbox there. When we look at yield across all the locations that had pressure, so we had 69 locations with pressure, but we're only able to get yield off 63. We can see that SmartStax Pro is out yielding all of our other hybrids, which greater root protection translates to greater yield in this data set, which is what we would expect. Now, we did look at yield at those locations that didn't have any corn rootworm pressure, just to see if there's, any effect on yield from the addition of that RNAi technology. And at least with these hybrids in this data set, we did not see a reduction in yield with SmartStax Pro compared to SmartStax or our competitor trait products there. So that was good to see as well, that even under no corn rootworm pressure, we're still seeing a yield advantage with the SmartStax Pro hybrids in this data set. So when we look at the summary of the data set, at those 69 locations that had injury, SmartStax Pro did provide 3 times better control than Chrome from Corteva. And SmartStax Pro and SmartStax hybrids were both more consistent in the control that they provided compared to our competitors in those really very high pressure feeding environments. And under corn rootworm pressure, we did see SmartStax Pro provide a 12 bushel advantage over Chrome products. And when we look at the performance consistency by technology here, this GTED, that would be that 0.5 threshold that I had mentioned earlier. We can see, 92 and 81% of our locations were below that threshold compared to only 46 and 19% when we look at Chrome. So, again, with this number, 92 and 81 are very good, but they're also not 100%, right? So again, it goes back to SmartStax Pro is very effective at managing corn rootworm, but it's also not the silver bullet, right? So we still need to be thinking about our different management tactics and tools that we have in the toolbox when it comes to corn rootworm. So these next couple slides are just some pictures comparing roots to some other trait technologies with the root scores. You can see some very good performance on SmartStax Pro, especially comparing to Chrome. And we can see, again, even under low pressure here, we're still seeing good performance from SmartStax Pro. But again, we want to make sure that we're utilizing these best management practices for corn rootworm, right? So again, like I've said previously, SmartStax PRO works very well. We have seen very good performance and protection in corn rootworm fields, but we need to think of it as another tool in our toolbox, and really need to think of corn rootworm management as a holistic approach. So kind of our best management practices would be, we emphasize crop rotation as the most effective practice, right? So corn rootworm larvae can only feed on corn. So if we rotate away from corn to soybeans or another non-host crop, that's the most effective way to manage a population in that field. Another way is use soil applied insecticides with a non-corn rootworm traded product. And so this will, the soil applied insecticides will provide some protection, so some yield protection and standability, but we're also going to be allowing, susceptible beetles to emerge from the non-corn rootworm traded to mate with any resistance beetles if that is a concern within that field. And then the other third option is, if we're still using a single traded product to manage corn rootworm, we really want to move away from those and switch to a pyramided product like a SmartStax or a SmartStax PRO, just because multiple modes of action present within the same hybrid are going to provide a lot better management and protection than relying on that one single trait. And so with that, that's what I had, Lauren. I can take any questions if we have them.
Yes, thank you. So I have a few questions and comments prepared, but if anyone watching also has questions, feel free to throw them in the chat and we can get to them too. But first of all, for that, I guess thank you for taking the time to present with us today and sharing your expertise on this topic. I mean, the Eastern Canadian Market Development Team has also been doing a lot of the root digs and running very, very similar trials. And in terms of trait efficacy, we've been seeing similar results in terms of, the advantage of a SmartStax hybrid over, competitive traits, and then the advantage of SmartStax PRO even over SmartStax. But in situations, in terms of recommending best management practices for growers, if you have a grower that's in a situation where their SmartStax or pyramided BT traded hybrid is starting to have, unexpected or unacceptable levels of injury on it, what's the best way for that grower to manage that? Should they, is rotation the number one strategy always?
Yeah, so if you've got a field that, we're planting a trait for corn rootworm in, and we're starting to see some trait performance issues, first thing is you want to understand, what's going on, right? So is it because we just have a lot of beetles and a very high population in that field, and so it's quote unquote overwhelming the trait, or do we potentially have a resistance situation going on in the field? So first thing I would do is go out to the field if it's around silking, see how many beetles are flying around. If you have some gooseneck or lodged plants, go to those areas, dig those roots up, see what kind of feeding you're seeing. If it is a traded product and it looks, and you do dig roots and it looks like heavy feeding, you definitely want to be calling either your sales rep or the agronomist or market development representative to get out there and do a little bit deeper dig into what's going on in the field. But if it is, decreased trait performance, the number one way to manage that's going to be crop rotation. So, I mean, I know some growers, at least in Iowa, in my territory, that have been corn on corn, 10 or 15 years. They do have issues with the SmartStax, where they're seeing heavy feeding, high populations. They've rotated the field to soybeans 1 year, and they've been able to go back to the field the next year with just a double pro without a soil applied insecticide. So that's how effective crop rotation is for managing rootworm. If you do have a situation where maybe it's a livestock producer, they need corn for the feed, then you have to get a little more creative, right? So if they're unable to rotate to beans, I know we've had some scenarios where growers will either swap acres with a neighbor. There is going to be a little bit of a yield benefit to switching to soybeans after being corn on corn so long. So sometimes that's an option. Another option would be, maybe if it's a 160-acre field, sorry for the acres, but if it's a 160-acre field, maybe we just split the field into 280s and rotate, the side that has heavy corn rootworm feeding, and then, do a vice versa back and forth. Final piece is we've also had some success with some growers looking at, other forage crops for feed. So I know some guys have looked at sorghum or Sudan grass, or triticale. Obviously, you'd want to check, make sure you can grow those crops in your area, and also be checking with a nutritionist, just to make sure that, if it's a dairy or beef or whatever, they're going to get what they need out of that. So requires a little more creativity, but essentially rotation is going to be your easiest and most effective strategy. So
Okay. Yeah, and those are similar kind of recommendations that we've been making, and we've been seeing similar success with growers that are able to rotate out, that even 1 year does help solve the
yep.
problem
Yeah.
pretty significantly. One of the other things we're actually seeing with growers, and this was something that was, I learned, I mean, I've been an agronomist in this territory for 3 years now, so this was something that was a little bit of a learning kind of in my first and second year. But sometimes what we see is, a grower that has a second year or a third year cornfield right beside a grower that has, 15 corn on corn for 15 years or more. But then the product performance call that we get is for the crop that's been corn for fewer years. And I was wondering if you've kind of seen the same phenomenon where it's not always your longest corn rotation that's having the problem, and what that could be caused by.
Yeah, so I have seen this occasionally, in the countryside. Um, my thought is, it it could be that the field that has been corn on corn for, that long term, it does have issues going on in the field, but the grower just isn't aware of it, or maybe he is, and it's not a problem. I work with some growers that have been corn on corn for 50 years. They know that the corn's going to fall over, but they're fine with it, right? So you could have that situation. The other situation that could be happening is if, depending on planting timing. So if the long-term corn on corn gets planted early and it's going to be producing a lot of beetles, but the rotated field, or the field that's only second year corn on corn, gets planted a little later, or silks a little later, that's going to create a trap situation where that silking corn, that silking later, is going to basically be a giant magnet for those beetles to want to fly from the long-term corn on corn field into the rotated field, uh, to lay their eggs. So, and it could be for all the other neighbors in the area too. So, that's why with a trap crop situation, you always want to try to prevent that, because you'd be recruiting beetles from your neighbors, and sometimes if your neighbors have problems, then they can become your problem too, which isn't always great. So
Yeah. So if you were a grower in that type of situation, getting out and scouting and looking at your crop, and kind of understanding where the beetle populations are, kind of right around actually this time, and even kind of into next week, would be a beneficial thing for you to be doing to make your crop rotation plan for the next year. So, logically, if in the summer which of your fields have the highest beetle populations, in I mean, at least the way I've thought about it is your fields in the summer that have the highest beetle populations, kind of in that mid-August, early to mid-August timeframe, would be your best candidates for rotating the next year if you're looking for fields to rotate.
yeah, I mean, the biggest conversation we've been having is like you said, we have to get back to a point where we're going out and actually knowing what's going on in these fields and understanding where the populations are at. And, I know that's not always what we want to be doing, right? So I know soaking corn is not always the funnest thing to walk through, but really that's going to be the best time to be out there to evaluate what kind of beetle pressure you have. Um, and especially if we're seeing areas of a field that are either lodged or gooseneck, we definitely want to be going there to dig to see if it's caused by rootworm, or maybe it was a poor root structure, or some other agronomic issue that caused that. But the the worst thing, and the thing that we don't want to do, is find out we have a rootworm problem when the whole field is lodged and it takes 10 times as long to combine, and then we're wondering if the trait performance is at issue as well. We want to try to change it up before we get to that standpoint. And that just all goes back to having a better understanding of what's going on in the field, field by field basis, so we can make those decisions.
Okay, cool. Is there any, if a grower wanted to go out and spray an insecticide for adult beetles, is there any advantage to doing that?
Yeah, so if it's done correctly, you can control adults and therefore reduce how many egg-laying females you're going to have, reduce how many eggs you'll have the next year. Um, I always I always caution growers that are thinking about doing this, as telling them the way the rootworm works is they're going to be emerging out of the soil for a 6 to 8 week period, with the majority of the beetles coming out over 4 weeks. So that's a long time period that you'll be having corn rootworm present in the field. And a lot of our foliar insecticides, the residual, if we're lucky, maybe 14 days at the most. And that's if we're really lucky. So if a grower is thinking of spraying for beetles as a management tactic, I always make sure that they're aware that they got to be out in the field scouting, right? Because we want to make sure that we're spraying when we have gravid females that are ready to lay eggs. And then after that first insecticide application, they probably need to be prepared to make a second pass, sometimes even a third, which again is going to require them to go back out into the field after the first spray. And once they start seeing populations increase again, they need to be able to pull the trigger. So it can be done, but it's not as easy as, throwing the insecticide in with the fungicide and calling it good, if that makes sense.
Okay. And yeah, and I mean, just as I'm thinking about it now, I mean, the economics of spraying an insecticide 3 times is likely going to be not great,
Right.
especially when that would be preventing the problem for the next year. Like, that won't save you any issues the year it's applied.
Right. Yeah. So you would, unless you have silk clipping going on, which, like I said, it can happen, but it's I would say it's relatively rare. But the only way you would be getting an ROI for that current year would be if you have the silk clipping going on. Otherwise, it's all about managing populations for the next year. And so, again, if you're talking spraying 2 or 3 times, you have the cost of the plane or the helicopter, plus, the cost of the product. It Most of the times it just pays to, either use a trait if you're not using a trait, or, rotate for a year, instead of, spending all the time and money on doing multiple applications. So,
Okay. Yeah, because that's a question that we get
mhm.
rather frequently, and we usually caution growers that, I mean, it's likely not going to pay for them, and it's probably not going to provide the result that they maybe want it to.
Yep. Yeah. I mean, you can do it, but you have to be willing to do it right and basically babysit the field, and be able to get the plane and the helicopter there, within a couple days of when they need to be there, instead of, I mean, I don't know how it is for you guys, but for us, it's, you get put on a list and it might be 3 or 4 weeks before they can get out there. So,
Yeah. And I guess just as we're talking about it too, thinking of pre-harvest intervals or anything as well. If you're going to have to consider that too. But anyway, the last few things that I kind of had to touch on was just around, the other questions that we often get is, is there any advantage to rotating competitive traits? Yeah, like if you wanted to go from a Chrome to a SmartStax back to a Chrome or have Duracade in there, is there an advantage to rotating BT traits, or essentially are they all the same mode of action?
Yeah, so they're similar modes of action, but different. That cry protein. Cry is crystalline. It's a crystalline protein. That's the shape, right? So without getting too technical, but basically the structure and shape of the proteins can be similar, and it's similar between the Cry3BB1. So originally the yield guard rootworm or the VT triple pro rootworm component. And then the two Syngenta traits, the mCry3A and the eCry 3.1. So the protein structure is similar enough between those three that you can get what we call cross resistance. So say I have a field that's resistant to Cry3BB1, and I want to plant the Syngenta traits in that field the next year. Because of cross resistance, even though that population has never been exposed to the Syngenta traits, I still could see issues with those. And that's just because of the way that the protein structure is. With the Cry 3435, we don't see any cross resistance there because the protein structure is different enough that we're not seeing any cross resistance there. So it's similar mode of action between all four BT proteins, but there's just those three where we see the cross resistance. So it again, it depends on what proteins are present within the trait package, on whether that's a good decision there or not. So.
Okay. But yeah, but I mean, even if you've developed resistance to one and you go in with another pyramided trait hybrid, you're still really down to one effective cry protein, which kind of sets you up for multiple resistance in near future.
Right. And, some trait packages, may have the same protein, and the same trade name, but it'll be a different event for the protein. And I'm talking about Chrome, right? So with Chrome and SmartStax, they both have that Cry3435 Herculex protein in there. But with Chrome, it's a different event, compared to the Herculex Cry3435 that we use in SmartStax. And the reason they went with that different event is because, it's easier for them to get the Herculex protein into more germplasm, and they don't see the yield drag that you sometimes see with the Herculex. But the trade-off for that is they do have lower expression of the Cry3435 in the Chrome. So that's another layer on top of the cross resistance. The added layer is, you may not always have the same expression level, even though it's the same protein and the same, trade name in there. So that would be another thing, another watch out.
Okay. Yeah. I mean, thank you. That kind of wraps up, all of the questions that I have. If there's anyone in the audience that has anything, feel free to throw it in the chat or comment your CCA number and we can submit for the CCA credits as well. I just didn't get that set up quite as quickly as I wanted to, but no, thank you again for joining us and taking the time to have this conversation. We're really excited about SmartStax PRO. Our field data has been phenomenal for efficacy. And yeah, we're just wanting to make sure that we use it properly and have it for a long time in the future. So
yeah, that's our goal too, is just make sure, it's another tool in the toolbox, but it's not the tool, right? So it's like you said, it's very effective and works very well, but we also don't want to rely on only one tool because, I mean, we've seen what's happened with our BT trait technologies, and that's it. We don't want you guys to break it before we do, and we don't want to break it either. So that's kind of, going back to those BMPs and having an understanding of what's going on in the field. That's the first starting point, and then we can start looking at the different tools we have available and which ones we want to go with. So, yeah.
Anyway, yeah, it doesn't look like there's any questions, so everyone can go a little bit early, and I'll just, thank everyone for their time today.
Yep. Thank you. And if you do have something that comes up, Lauren has my contact info, so feel free to give me a shout and we can talk through it. So, yeah, thank you.